I'm Rosalind Adamson. I work at the Structural Genomics Consortium in Oxford in the UK. I'm working on two proteins that are involved in Huntington's disease. These proteins are called the ATM kinase and the KEAP1 CUL3 complex. We're funded by CHDI, the Cure Huntington's Disease Initiative, and they are very interested in discovering the various ways that Huntington's disease is caused and how we can find various compounds in order to inhibit or cure the disease. The SGC has an open access policy, and all of our structures and information about the kind of research that we do is openly available in the PDB or on our website. Huntington's disease is a neuronal degenerative disorder that is generally late onset after adulthood and can take up to 20 years from initial diagnosis to death. The disease results in cells dying in the brain, and this causes a number of problems with movement, with speech, with memory, and with coordination type problems. The disease results from what we call CAG repeats in the Huntington protein. These are repeats in the DNA sequence of the Huntington protein, and the more repeats of them there are, the sooner the disease is diagnosed and the more dramatic the decline in the patient. Oxidative stress happens in healthy people as well as people at risk of Huntington's disease. Oxidative stress is a result of various cellular processes in the body, including the processing of glucose, for example. Smoking and other added toxins in the body can also lead to oxidative stress. Huntington's disease sufferers have an inherited inability to deal with controlling the oxidative stress imbalance that result from these stresses. Oxidative stress can lead to DNA damage, and if the various protein complexes that are involved in DNA damage control cannot repair the damage, cells die in a process called apoptosis. This leads to neuronal degeneration and can lead to the disorder of Huntington's disease for people who have the mutation in the protein. It's a degenerative condition and it currently has no cure. We may be working on potentially hundreds of proteins every day in the lab.